Okay, welcome back. Here we are moving on to the senses. Boy, well, how time flies in the summer when we're having fun. So, tonight, what we're going to talk about in this lecture presentation, we're going to talk about the eye and ear. And we're going to just go right down the checklist, and we're going to tell you there's some things that we can cross off. But what we're going to do is we're going to look at the human eye right now. So, when we look at it, there are six muscles that control the movements of the eye. And those are your rectus muscles. So you got medial, lateral, superior, and inferior. And you have two oblique muscles. And then the last muscle in your checklist, the superior levator palpebrae, that is the muscle that controls the upper eyelid. That is what will open the eyelid when that muscle contracts. So if we were to start to come right on into the muscles right here, this first muscle right here, this is the lateral rectus muscle. Now, if we take these other muscles off for right now, what we will be able to see is we will be able to see the medial rectus here alongside the nasal cavity. And then running underneath, even below right here, running right in through here, this is the inferior rectus. Okay? And then when we put the eye back together again here, we have this muscle right here, and we got to look really close because there's two muscles that sit on top of one another. This muscle right here is the superior rectus muscle. So again, when we're looking at the rectus muscles, they're coming straight into the eye from all four sides, top, bottom, right, and left, coming straight into the eye or attaching to the eye. Now, the oblique muscles come into the eye at an angle. So when we look at this, we're going to remove the superior rectus, and we're going to come back and we'll explain what this muscle is right here. But what we have this muscle right here, and if we follow its course, we will follow the muscle down, and then we will see it make an angle and come and attach into the eye right here. So what we have right here, this is the superior oblique. And what it helps us identify the superior oblique very easily is it passes through a cartilaginous structure called the trochlea. So if we look right in through here, this structure right here, this is the trochlea. We can see the superior oblique come, and then the superior oblique passes through the trochlea before it attaches to the eye. And then as we come down, what we can see right here on the very bottom of the eye that almost forms like a sling or a hammock on the inferior portion of the eye, that's the inferior oblique. So again, if we want to really just look at it, the inferior oblique will be here, the superior oblique will be running here on top of the eye. So they come in at angles. Whereas the rectus muscles will be coming straight in from the back. And then the last muscle, again, is the one muscle that does not control the eye, but that is going to be the superior levator palpebrae. And that muscle sits right on top of our superior rectus. And as we follow it out, we see it not attached to the eye, but it comes out and attaches out here to the eyelid. And so when that muscle contracts, what that's going to do is that's going to pull the upper eyelid upward. Okay, moving down into the checklist, we're going to talk about the lacrimal apparatus. And the lacrimal apparatus is what produces the tears for our eyes. And again, the purpose of tears is not so much when we're upset. That's just a nervous system response. The gland becomes very active and produces tears. But under normal circumstances, what the tears in our eyes are there for is to keep the eye clear of waste and debris. Anything that gets in our eyes, think about it, anytime you get anything into your eyes, you naturally water up, and that's the body's mechanism to try flushing the eyes out. So the first structure on the apparatus is this structure right here, and this is the lacrimal gland. And that's going to be basically found superior and lateral to the eye. So when tears are produced, the tears will move from lateral to medial in the eye, and that ensures that the entire eye gets basically saturated with tears to flush anything out that may end up on the surface of our eye. The lacrimal ducts are how the tears reach the eye surface. And if we come right in here on this structure right here, these white lines on this model right here, those are the lacrimal ducts. So there's the lacrimal gland that produces the tears. Then the ducts lead the tears right to the surface of the eye. And then what happens is, and if you think about when you're crying, where do the tears usually start to fall down your face? Usually on the medial side, because that is where the collection point is. That's where the tears are collected. So the first two structures are the superior and inferior lacrimal punctum. And what we're looking for in the medial corner of the eye are these two small openings. And they are very small, there's no doubt about that. But there's the superior lacrimal punctum, there's the inferior lacrimal punctum. And then those structures lead to these two passageways right here, the superior lacrimal canal, the inferior lacrimal canal. And then what ends up happening are the tears then will be collected by this structure right here in the medial corner of the eye, and that is the lacrimal sac. And then the lacrimal sac runs inferiorly into this opening right here, or this larger area right here. This would be the nasal lacrimal duct. So again, think of, there's the little dilation right there, that's the lacrimal sac. And then this inferior to it, the lacrimal duct, the nasal lacrimal duct comes and it empties into our nasal cavity, which will explain why when you have a really good cry going, you usually have an associated runny nose. That's just the excess tears. The conjunctiva. The next structure is the conjunctiva, which is a membrane that lines the inner surfaces of the eyelid in the anterior portion of our sclera. And again, our sclera are the whites of the eye. Make the note, the conjunctiva does not cover the cornea, which is the transparent window of the anterior eye. But so if we take this model off right here, the conjunctiva is this structure right here in pink. And it comes all the way around, lining the eyelids. And then where that membrane would wrap around would be this portion of the sclera right here, again, which are the whites of the eye, and this portion of the sclera. The conjunctiva does not cover the cornea right here. And again, the whole purpose of the conjunctiva is to produce a watery secretion to help reduce friction when we open and close our eyes. And again, anybody who's had dry eyes at one time or another, we all have, that tells you that your conjunctiva is not producing the secretions that it should. And one way that we combat that, eye drops. The eye drops act as the secretion that the conjunctiva puts out. And then last but not least, the next structure in the checklist is the lacrimal caruncle. The lacrimal caruncle is the bump in the medial side of the eye. And again, there are sebaceous glands in there to produce a little oil to help keep, again, the surface of the eye moist, the eyelids moist. But again, over time, if we're overproducing that oil, what it will do is it will oxidize to air. And that's when you wake up in the morning and you got all that crust, or as other people call them, the eye boogers. So that is the lacrimal caruncle. So as we move in, there are three layers to the eye. The first one being the fibrous tunic. The next layer in would be the vascular tunic. And the innermost layer would be the nervous tunic. And again, inside each layer, there are specific structures that you need to know. Now, for the fibrous tunic, the outer layer of the eye, that will be the two structures we need to know: the cornea and the sclera. And again, right here, if we just take the eye out right here, just take the eyeball out. Well, the bottom half will stay in, but the white of the eye, that's the sclera. And we'll notice the sclera goes all the way around. So that is sclera. And then the anterior transparent part of the eye right here, this structure right here, this is the cornea. The importance of the cornea is what allows light to enter the eye. That's why it's important that that area is transparent. If it was made up of the tissue that makes up the sclera, light rays would hit off of it and we would be blind. So it's important that the cornea it remains always transparent. And those are the two structures in the vascular or in the fibrous tunic that you're responsible for. The next layer in then of the eye would be the vascular tunic. And what we can see, this brownish structure right here, that is part of the vascular tunic, and that is the choroid. And if we can see, the big key to noticing what the choroid contains blood vessels, and this is going to supply the eye wall. So the darker area right inside, just deep to the sclera, is the choroid. Also, what we will find in the vascular tunic, we will find the iris, which is the colored portion of the eye right here, and in this one it's yellowish gray. So anybody with yellowish gray eyes, don't worry, you're not left out. They made a model for you. And then the opening right in the middle of the iris is the pupil. And again, that allows light to reach the back of the eye even further. Okay, so that is the choroid, iris, and pupil. And then number 4 is the ciliary body. Ciliary body is made up of muscles. And basically what its purpose is, the ciliary body is going to contract and relax, and by default, that is going to change the shape of our lens, which we'll explain a little bit more in detail as we move in here. But our lens is what brings images into focus for us. So if we look right here, this structure right here in yellow, right in front of the, well, no, that's not it. So right back here. So right here, this dark structure right here, this is the ciliary body that comes all the way around. And again, it's made up of tiny processes, and all together, that's the ciliary body, and that's what we want you to know it as, the ciliary body. And again, what sits right in the middle of the ciliary body is the lens. And there's our lens right there, sitting right there. So the lens is just posterior to the pupil and the iris. And as we go down to the checklist, and I want you to jump down to other important structures right now, down in your checklist on page 32. These white structures right here that we're looking at, those are suspensory ligaments. The ciliary body never directly attaches to the lens. There's this little junction right here, and the little junction that connects the ciliary body to the lens are the suspensory ligaments. So when the muscle within the ciliary body contracts or relaxes, that's either going to tighten the suspensory ligaments or loosen the suspensory ligaments, and that's going to cause the lens either to thicken up or flatten out. And that happens all day long in our eyes when we're looking at things at different lengths, different distances, different text sizes, but those are the suspensory ligaments right there. Now we have another view on where we can show you the suspensory ligaments, and again, this is in the APR picture, so it's very important that we look at this. You're looking at your APR as you're studying. So right here, as we come in, we can see, when we look at a cross section, we can see the iris right here, and what's the hole in the middle of the iris? That would be the pupil. We move posterior to that, there's the lens, and then now we can see these tiny little hair-like structures, are these little guitar strings running and attaching to the lens. Those are the suspensory ligaments. You follow those out, and where would they go to? The ciliary body. Okay, so again, then it's going to change the suspensory ligaments, and whether how tight they are, how loose they are, is going to change the shape of the lens. Okay, and then finally we're going to move into the we're going to move into the nervous tunic, and the primary structure in the nervous tunic is the retina. The retina is the actual nervous tissue of the eye. And so right here is the retina would be back here when we put the eye together. So again, if we put this together like this, we take off the sclera, that exposes the choroid. We take the choroid off, that exposes the retina. Now within the retina, there are two types of photoreceptors which are stimulated by light, and those are what generate the nerve impulses for our sense of vision. And again, going back to our nervous system, where was our sense of vision interpreted at? It was in the occipital lobe of the brain. But so here is the retina. What are the photoreceptors found in the retina? The rods and the cones. And again, the rods are what are stimulated more in dim light, and rods help us make out shape when there's very little light. And now the cones, what the cones are responsible for is for us to be able to see color in detail. And then the next structure is the macula lutea. The macula lutea is a circular region at the back of the retina, and here it is right here. And sometimes it is referred to as the yellow spot. Now we got to add, usually this area turns yellow once a person is dead. Just the pigmentation changes here, and that's what causes that area to turn yellow. But it's that circular region right here, the macula lutea, and then smack dab right in the middle of the macula lutea, we have the fovea centralis. The fovea centralis is just a small depression in the middle of the macula lutea, and what it contains is it contains the greatest concentration of cones that we will find anywhere in the retina. So, and actually this is what the lens and what the eyeball is trying to focus the image of what we are looking at over directly, right over the fovea centralis. Because of the large concentration of cones, this is the area of our greatest visual acuity. This is where we're going to be able to see things the most clearly that we ever will be able to see them. And then there's another region right over here. If we move over this circular region, and we can see blood vessels coming out of that area. And what that is the optic disc, or so you can call it the blind spot. Now the importance of this area is this is where the optic nerve meets the retina. So if we flip this back to the posterior side of the eye, there is the optic nerve. Now when the optic nerve comes into the eye and it merges with the retina, what is formed here is the optic disc. Now one thing that's very important right here is that there are no rods or cones in this area. So even if an image and light were to strike that area, we would not be able to see it because there are no rods or cones there to be stimulated. And so again, so you're looking for two circular structures on the very back of the eye. The big one right here, macula lutea. What is the small depression smack dab right in the middle? The fovea centralis. What is this circular region right here with a large number of blood vessels coming out from it? That would be the optic. That would be the optic disc. Also in our eye, there are two main chambers in our eye. You have the anterior chamber and the posterior chamber. The anterior chamber extends from the cornea to the lens. And what it's filled with a solution called aqueous humor. Aqueous humor is a watery substance that is constantly being produced by our eye. And what it is, it's there to help keep the internal structures of the eye moist. And then what we can further do, because again, everything in A&P we can break down further and further because you guys don't have enough to know already. You can break the anterior cavity down into two chambers. We can break them down into the anterior chamber and the posterior chamber. The anterior chamber extends from the lens, or from the cornea, excuse me, to the iris. So from the cornea to the iris, this is the anterior chamber of the posterior cavity. The posterior chamber extends from the iris to the lens. So again, it's just this little opening right here. Altogether, what is that space? Anterior cavity. So again, if we just said name this space, name this cavity, anterior cavity, name this specific space, we would want anterior chamber or posterior chamber. And then the last structure, the other structure is the posterior cavity. Posterior cavity is much larger than the anterior cavity. And basically it extends from the lens to the retina. And here it is right here, folks. There is the posterior cavity of the eye. And what it is filled with a gelatin-like substance, which is called vitreous humor. One of the big things about the vitreous humor as compared to the aqueous humor is, besides its composition, is once we reach full maturity, we do not produce any more vitreous humor. So once we reach full maturity, we're done producing vitreous humor, and that's why it's so important that we maintain and we keep our eyes safe, because if we're ever to lose vitreous humor, that can alter the way that the eyeball functions. So again, posterior cavity, what's the fluid filled in it? Vitreous humor. Okay, so moving down in the checklist, underneath the cavities and chambers, at letter B, the canalus schlem. What the canalus schlem is, the canalus schlem are openings at the junction of the cornea and the sclera. And what it allows for the drainage of aqueous humor from the eye. So if we come up onto APR picture again, what you'll notice a small opening right here and another small opening right here. That's the canalus schlem right there. Those are the openings right there that shows you the canalus schlem. Also, if we look on this eyeball model right here, in pink right here, those openings are trying to represent the canalus schlem. And again, what is it there for? It is there solely for drainage purposes of the aqueous humor from the eye. And then down at the bottom of your checklist on page 32, folks, the other important structures, we already covered lens and suspensory ligaments just earlier. And Mr. Fogel has already covered these with you, some of these, optic nerve, optic chiasm, optic tract. He covered those with you in the nervous system. But we can review those real quick. So again, we're on the inferior surface of the brain. We can see the olfactory bulb, the olfactory tract. We can see the mammillary bodies right here. And now again, what we can pick up is we can see the optic nerves. Now these optic nerves have been cut, but that's okay. And the optic nerves come back together to merge to form the optic chiasm. And then from the chiasm, we have these projections right here. They come back, and those are the optic tracts. Now, the optic tracts then will synapse. They're the nerve fibers that are traveling then, those will synapse with these structures right here, which are called the optic radiations, or you see in parentheses, projection fibers. Whatever you want to go with is fine by us. But these are the structures that are going to take the final nerve impulses back to the occipital, the visual cortex and the occipital lobe. We can see that coming right here. We can see them coming right here. So again, optic nerves merge to form the optic chiasm. The optic chiasm then sends off the optic tracts, and then the optic tracts and the nerve fibers that they contain synapse with the optic radiations. And the optic radiations are what are going to bring the final nerve impulses back to the occipital lobe. Okay, so what we're going to talk about are a few special tests that we can do for our eye, just to test someone's sense of vision. This card that I'm holding up right here, this is the test for the what we can test the optic disc with, or we can test our blind spot. We just draw a plus and a dot on an index card, and what you would want to do is you want to cover one eye up And you want to just focus on the plus sign. And as you move that card towards your face, for a brief second, the dot will disappear from our vision, even though it's right in front of us. And then it will reappear as you continue to move it closer to your face. What that is telling us is that dot at that one particular moment where it disappears has passed over the optic disc. And again, why don't we see it? Well, there are no rods or cones to stimulate. And that is why when that dot passes over the optic disc, we don't see it for that brief second. So that is, what does this index card test for? The optic disc. What is missing at the optic disc? What receptors are missing at the optic disc? Rods and cones. Okay, so what we're testing now, folks, we're going to move right down the checklist and we're going to test visual acuity. And I feel pretty comfortable that you are all familiar with this chart. And this chart's name is the Snellen chart. And what does this test for? This tests for visual acuity, how clearly we can see something. So as you come down, the term emetropia, if you see that, normal vision, which is 20/20 vision, that's what's considered to be normal, 20/20 vision. And we'll explain what 20/20 vision means and some other visions. So again, right here, this is the Snellen chart. What does it test for? Visual acuity. So as we come down, if we look at our test results, when we look at a test result of 20/40, so right here, 20/40, what does 20/40 mean? So what that means is what the test subject sees at 20 feet, the healthy eye sees at 40 feet. So anytime the bottom number is higher, that's telling you what the healthy eye can see. Okay, so 20/40 vision. What the test subject sees at 20 feet, the healthy eye sees at 40 feet. So that person would need glasses. Now again, where the 20 comes from is that's the distance every test subject stands away from this chart. It's always tested at 20 feet. Now also too, the next one in your checklist, what's 20/15 vision mean? 20/15 vision means what the test subject sees at 20 feet, the healthy eye or the typical eye sees at 15 feet. So what does that mean? The test subject has better vision than the average or the healthy eye. So those are the test subject has better eyes at that point. So we do want you to know that. We do want you to know how to read the Snellen chart for the third lab practical. So again, what does 20/40 vision mean? What the test subject sees at 20 feet, the healthy or typical eye sees at 40 feet. Okay? And then the last one that we're going to make you responsible for is the test for astigmatism. We're not worried. We're not going to hold you responsible for the chart for the astigmatism. But what astigmatism is characterized by is blurry vision. And why the vision is blurry of somebody who has astigmatism, there's an abnormal curvature of either the lens or of the cornea. And those are the two structures. You could have one structure could be affected or both structures could be affected. And the reason why those are the two big structures in astigmatism that we're worried about, they are the structures that refract or bend light rays when they come into the eye. So if they have an abnormal curvature to them, the way that the light rays get bent or refracted, and we'll talk more about that in lecture, that affects the way an image is focused on the back of our eye. And then near point accommodation, you are not responsible for, so you are welcome.
What we're going to jump into now is page 35 of the checklist. This is going to be the special senses, specifically the ear is what we're covering here. Now we have a lot of amazing models here in class to utilize. Unfortunately, I wish we could put them in your hands, but we're gonna be giving you a visual stimulus, hopefully, which will cover that pretty well for you. Do not resist the temptation to jump on APR, so get on there, start looking at some of the structures on your own. But we're gonna show you this little left ear model here that we have in particular. So simple enough, external ear, what we see on the outside. The external ear is the portion, right? We can go ahead and flip our ear and it pops right back into place. We have the pinna. We also call this the auricle, or the outer portion of the ear that we're gonna see through here. So, right, you can stick our finger in there, clean some of the earwax out, whatever you wanna do. Now, if we look within the ear, all right, what we're seeing now is known as this opening here, the external auditory meatus. Now, you can use any combination of the words that we have there. We just asked you that we use the full terminology: external auditory meatus, external auditory canal. So simply enough, the outer opening into the ear canal itself here. Now, what we need to jump into now is the next portion, the tympanum. Now, the tympanum is going to be known as the eardrum. If we look in through this canal here, so we move into the external auditory meatus, we move in here and we come to this portion right through here, this drum-like structure. This drum-like structure is going to be the tympanic membrane, or you can also call that the tympanum. It pops up pretty easily, so if you stick a Q-tip in your ear, try to resist that. But you can poke that very easily and cause some kind of, just damage in general to this tympanum. So there we go. Now we're approaching to the middle ear portion, deeper structures. Now when we're getting in deeper, now we're seeing that actual bony structures, which we talked about with the skeletal system. And just to break it down, to show you on a visual standpoint, the three bones, the ossicles. These bones were known as the malleus, the incus, and stapes as well, all three bones. Now they're gonna connect to one another via the tympanum. So if we start at the tympanum here, we go ahead and take that apart here, we can see all three bones in the row. One bone, two bones, and the third bone is right there. So if we go ahead and put this all back in place, we can kind of see how they all connect to one another. We have the malleus connecting to the incus then attaches to the stapes, and that in turn is going to attach directly then to the inner ear, which we'll get to here in a moment. Now letter B is very important. This is the eustachian tube. The eustachian tube is going to be the structure which allows for drainage of the ear. So if we pass where the tympanum would be, so we have the tympanum sitting here, we remove that structure, we're going to see a tubular structure working medially and inferiorly. All right, so this is going to be the eustachian tube. It's connecting the nasopharynx with the middle ear all the way down to what we say is the throat. All right. Take this note though too, the middle ear is separated by a inner ear by a thin bone, a partially bony structure, which contains the oval window and the round window. We're going to get those structures momentarily. Inner ear though, that is our model here. Okay. Now what we like to do is take this model and blow it up to give you a better perspective of what all these different structures are. So I'm going to take that away, and I'm going to give you this gigantic elephant model. Well, maybe it's even bigger than elephants, who knows? But anyway, it's a blown up version of our human inner ear. All right. Let's go ahead and cover some of those basic structures that I just touched on in the middle ear, the separation points. The round window and the oval window, to be specific, first and foremost, okay? So if we look around at this structure, it is bony in nature, very solid in nature. But if we go ahead and turn it to the underside, we can see two structures here. We see an oval-looking structure, and we see a circular-looking structure. What we need to know is that the oval window, okay, is where the stapes is going to sit. Okay, the stapes is going to rest directly against this, and in turn is going to cause vibration then into this inner ear model. Now, if we give you the smaller version, right, we can see very well that the stapes is hanging here, and it connects directly then in turn to this oval window structure. So we're going to put that down. I'll stick to the big one. The bony labyrinth is the next section that we're going to look into, and the bony labyrinth are the structures which are part of the bony labyrinth, all the bone-looking structures. Okay, all these bone-looking structures in general are the bony labyrinth. But we're going to break those down into separate portions. We have three major portions, and these are going to be the semicircular canals, vestibule, and cochlea. So if I go ahead and take my hand on, put it on these little rings, you might say, these 3 rings that we see here, these are the semicircular canals of the bony labyrinth. 1, 2, and 3 semicircular canals. If we move to the middle portion, where I can grab at the central portion here, this is what's known as the vestibule, where we see the semicircular canals come into the contact with the middle portion of this entire bony labyrinth. And then if we extend outward then to this more, I guess you could say, look kind of like a snail shell structure, this would be the cochlea. All right, so there's the entire bony labyrinth. Back into our inner ear model. And what we can see on the outside, again, is this bony labyrinth. We had the semicircular canals, we had the vestibule, and we have the cochlea. Now we want to imagine if we chipped away the bone, we can see more underlying structures. And these are going to be consisting structures called the membranous labyrinth. So the membranous labyrinth is deep within the bony labyrinth. So we chip away the bone, right? Chip away the bone, that's what we can see here in gray and pink as well. Let's break down all these structures one by one. Number 1 is your semicircular ducts. The semicircular ducts, the note for this is that you're going to find these tubular structures within the semicircular canals. So if I take my probe here, right, we can see within the semicircular canals, we have this gray structure here, and this will be a semicircular duct. We have one right through there, and if we can imagine 2, then we can see how another one will come through here. And I know it doesn't show it quite well, but we would imagine that another one will come right through here as well. So we have 3 semicircular canals containing gray semicircular ducts. Okay. Now, if we go down to number 2, ampulla, and ampulla are a bulb-like structure at the tail end or merging point, you could say, of the semicircular ducts. So if we follow a semicircular duct downward towards the vestibule, we will see an ampulla. We have another ampulla, and we have another ampulla right there. So we'll see 3 of those associated with the semicircular ducts. Again, in gray is our focus here. If we move downward, we have utricle and saccule. Okay, so these are found within the vestibule. These 2 gray structures, again, the utricle, upper portion, it's a large membranous chamber within the vestibule. If we look just below that, we have the saccule. This is the lower, smaller membranous chamber within the vestibule as well. Last but not least is going to be the cochlear duct. We'll cut to that here in a little bit, but we wanted to tell you what are these fluids that we see surrounding the membranous labyrinth structures. So again, the membranous labyrinth, gray. But if we look just outside of it, we see this pink hue right through here. This pink hue is perilymph, perimeter, right, outside. Perilymph is in pink. This is the fluid that surrounds the entire membranous labyrinth structures. What we need to know that is within all of these structures now, within the membranous labyrinth structures, deep within them, we will find endolymph. Endolymph is fluid inside of all the membranous labyrinth structures, and we'll get to those along with the cochlear duct shortly. Moving on in your checklist to the internal structures within the cochlea, okay? We're gonna take this model, we're gonna pull it apart first and foremost to look deep within. And what we'll notice is that with that snail-like structure, you know, we see tubular-looking aspects. We see ducts running through. And if you imagine that these ducts run all the way through and around and keep circling all together and go to this most outer tip, which we'll define here in a little bit. So if we take this model, this segment right here, right, and we take that and blow it up, here's the large version of what we're looking at. We can see a pink, we can see a blue, and we can lastly see a green portion going along with this model here. So let's break those down one by one. The first one, the upper pink chamber, is going to be known as the vestibular duct. The vestibular duct is filled with perilymph, and it's going to extend from the oval window all the way down to the what's known as the helicotrema. The helicotrema we're going to define as this most outer tip here. And if we show you that on a different model, much better model here, you can see actually how the chambers connect at that most outer tip, the helicotrema. But going back to our model, again, vestibular duct, this upper duct. Also on this model here, we're going to define that as the pink duct that we see here, the more upper one. So if we see the vestibular duct, we can see another structure called the vestibular membrane. The vestibular membrane is going to be separating, okay? Creating a separation, you could say, of the cochlear duct from the vestibular duct. So if we look at the vestibular membrane, we can look just below that, and we're going to find what is known as the cochlear duct, sometimes known as scala media. Okay. But this chamber is going to extend from the helicotrema to the round window and is also filled with perilymph. Oh, I'm sorry, endolymph, my fault. Endolymph, said that wrong. So again, perilymph on the outside here within the vestibular duct, endolymph within the cochlear duct. Let's move on down to one more duct, and this is going to be the tympanic duct. The tympanic duct is green on the model. But if we blow that up and look at the large model here, it is this lower chamber that we see. Tympanic duct. Okay. The tympanic duct is filled with perilymph, just like the vestibular duct, also filled with perilymph. The difference is that with the cochlear duct, this will be filled with endolymph instead, so deep. Now we see a couple other structures associated with this cochlear duct here. The first one that we want to see, and take note of, I guess you could say, is the organ of Corti. The organ of Corti is this large bulbular looking structure that sits on this membrane here called the basilar membrane. So I like to say the basilar membrane is the base in which for the organ of Corti to sit. Okay? And you can see some little hairs extending off the organ of Corti. And these little hairs cause a little vibration, and it's going to cause this membrane to move. This membrane is the tectorial membrane. And as the tectorial membrane receives information, we extend outward, and we can see this blue portion. Anytime we see a blue portion connecting to this yellow portion here, okay, these are always going to be associated with the vestibulocochlear nerve. So if we follow this outward in yellow, vestibular cochlear nerve running outwards from the entire cochlea. The other portion that we need to go ahead and touch on now was the round window. So we had found the oval window before where the stapes attaches then to the vestibule. We move just below it and we can see the round window. The last figure that we had talked about one more time, helicotrema, the most upper tip portion of the cochlea here, helicotrema. Now at the bottom of your checklist it says, what is the difference between conduction deafness and sensorineural deafness? Well, we got to think about it this way. Conduction deafness is focused on physical properties. Can vibrations be felt, or in a sense, I guess you could say heard in this case? Okay. So if we look at our checklist, okay, we got to think of structures that are physical that could be affected, broken, or even punctured in that case. So if we move over to our eye model here, okay, I had talked about putting a Q-tip in your ear. We can easily cause conduction deafness if we were to puncture the tympanum. Also, we could have a breakage or separation of these ossicles. So the ossicles may not be vibrating to actually affect then the inner ear model. Last but not least, the only other thing that could be happening then too would be, what would it be? Well, that'd be about it really. Well, the cochlea itself could be damaged. You could have any damage to the entire bony labyrinth. So if the bony labyrinth is damaged, we would have conduction deafness. If we have damage to the tympanum, we would have conduction deafness. If we had damage to the ossicles, we would also have conduction deafness. Now on the other end, we have sensorineural damage. Sensorineural damage is damage to things such as like the vestibular cochlear nerve. So again, if we look back at our bony labyrinth, we can see the vestibular cochlear nerve here, leaving the cochlea. If there is damage to this, we could have sensorineural deafness, that information that could not be sent to the brain itself. We could also have damage to the organ of Corti. The organ of Corti, say for instance you listen to your music too loud over time, you will damage your organ of Corti, and no longer will this cause enough stimulation to cause the tectorial membrane to actually pick up that information to that vestibular cochlear nerve. And then last but not least, the auditory cortex of the brain could be damaged. So come with me on a field trip. We had our color brain model, and our auditory cortex could also be damaged when we're looking at sensory neural damage.
Okay, and so what we're going to begin right now is the endocrine system. Now, it's important to note for our lab purposes, all we're gonna have you do is identify the endocrine gland or the endocrine structure. We're not going to ask you what its function is. We'll talk more about that in lecture. So a lot of these structures you already know because we've already covered those, but so these are just solely structures of the endocrine system. So the first one is the pituitary gland. Now on this model right here, you guys are familiar with the pituitary gland now because Mr. Fogel has gone over that with you in a previous lab video. The pituitary gland is found on the inferior surface of the brain. And what is it held to by, what is it held to the brain by? And that structure is the infundibulum. So we can see the infundibulum right here, holding the pituitary gland to the brain. Now we're going to further subdivide it. The pituitary gland can be further broken down into the posterior pituitary gland and the anterior pituitary gland. And what we do is we do have a picture of this right here. In this picture right here, this is showing this. So we can see the infundibulum coming down here. And then there is the anterior pituitary gland, and make note the anterior pituitary gland is larger than the posterior pituitary gland. And again, when we're in lecture, we'll discuss more about what these two specific regions of our pituitary gland does. Okay, and then while we're at it, here's the infundibulum, and what does the infundibulum attach to? Right here, the hypothalamus. Okay, and the hypothalamus does play a major role in the endocrine system functioning. So again, if we were to split the brain up right here in half here, again, here is our diencephalon, which again, the superior portion is the thalamus. And then as we come down, here's the hypothalamus. And then yes, we can see the infundibulum leading right to the hypothalamus. And then finally, while we got the brain open right here, this little gland sitting right here, just superior to the colliculi right here, there is the pineal gland. And so if we pull out the brainstem model yet again, once again, we can see our superior colliculi, the inferior colliculi, and then this little pink structure that's sitting right above those. That is the pineal gland. The thyroid gland, again, found in our cervical region, just inferior to your larynx. So if you find your larynx and move slightly down, you will be eventually coming into contact with your thyroid gland. And here is the thyroid gland right here. We can see it come all the way across, just inferior to our larynx. And then on the posterior surface of the thyroid gland, if we turn this around, we can see these red dots right here. Those red dots are representing the parathyroid glands. Parathyroid glands are their own individual glands, but they are found embedded within the thyroid gland's tissues. So you really have to watch if we put a picture like this on the test. Or do we want the thyroid gland? Or do we want the parathyroid glands? So again, look closely at what we're asking on the question. Moving down, the thymus gland. The thymus gland, again, is found superior to the heart, folks. So here is our heart. We just move slightly superior to it. Here is the thymus gland right here on Thin Man right here. Again, there's our larynx. What's just inferior to the larynx is the thyroid gland. And again, I know that can get a little confusing, thyroid, thymus, but we have to know that. Again, the thymus is just superior to the heart. The thyroid gland is just inferior to the larynx. The next structure then is the adrenal gland. And again, the adrenal gland, folks, is found right here, sitting right on top of our kidneys. So that's the adrenal gland. Pancreas, again, pancreas is found on the posterior surface or the posterior region of the stomach. So again, there's our stomach. We flip this over, and then there is our pancreas right here. posterior to the stomach. Ovaries and testes, again, I think we know the difference between them, but we'll go into them anymore. Anytime we see something hanging in this general vicinity, folks, pretty good chance that that's a male. And there's our testy right there. And here's the female reproductive system. We can see the urinary bladder right above it. That is the uterus. So we trace the uterus out, and then we follow the fallopian tube down and around, and right to the end right here, there is the ovary. And with that being said, that is the endocrine system, and that's as simple as we're going to make this on the lab test. We're not going to ask you for structure, for the actual functionality of these glands. Again, we will discuss that more in lecture, what they're responsible for producing.